Colorectal cancer, NanoCircDx.
Hello and welcome to NTHU News Channel, bring you the latest news around the globe. My name is Kira. Our story today is about the importance of early detection of colorectal cancer. Colorectal cancer ranks as the third most prevalent cancer worldwide, contributing to 940,000 deaths and 10% of cancer-related deaths globally. Due to colorectal cancer, there are over 10,000 annual diagnoses and more than 5,000 deaths in Taiwan. Colorectal tumors may cause bleeding during bowel movements, leading to blood particles adhering to the stool surface. The primary diagnostic tool, named fecal immunochemical test, FIT, it detects hidden blood in the stool. FIT boasts a sensitivity of 79%, but it still carries a 21% margin of error and has drawbacks, including dietary restrictions and specific medication considerations. Today, we have the privilege of interviewing the NTHU Taiwan iGEM team. Let's welcome their marketing manager, Yvonne, to talk about their development.
Hello, Kira. Hello, everyone. I'm Yvonne, the marketing manager of the NTHU Taiwan iGEM team. Our team has been focusing on the significance of early screening for colorectal cancer. We noticed that there's no effective method for its early detection so far. To fix this problem, our team has developed a remarkable and innovative device for the early detection of colorectal cancer called NanoCircDx, which has numerous advantages, including convenience, accuracy, minimal invasiveness, and affordability.
So, Yvonne, are you certain there is a genuine demand for NanoCircDx in the market?
Well, yes. According to our public questionnaire, 85% of respondents consider colorectal cancer serious and common. 96% of respondents are willing to use our product due to its convenience, accuracy, and low cost. Our team has also applied a full-piece marketing strategy to analyze our product. According to a report by Markets and Markets, the global cancer diagnosis market will reach a value of $29.6 billion in 2026. Our product harnesses the power of innovative liquid biopsy technology. Based on our public survey, this method effectively solves the shortcomings that the general public often associated with the existing methods, such as inconvenience and invasiveness.
How about the price? Does your product have any advantage over traditional one?
In terms of pricing, our product is more affordable compared to the traditional colonoscopy. Patients only need to cover the registration fee and associated blood testing costs. Also, through the use of fully automated equipment, our product can effectively reduce hospital labor costs, alleviating the burden on the healthcare system. As we continue to develop and refine our product, we plan to cooperate with local clinics and healthcare institutions on promotion and venue. We aim to make a meaningful impact on the field of detection and ultimately save lives.
So, there seems to be a genuine demand for the market. I wonder, how does NanoCircDx work?
For this question, let's call out our WebLab member to introduce this.
Hello everyone, we are members of WebLab. My name is Cindy.
My name is Anne.
Today we are going to introduce our project. Recent detection methods, colonoscopy and fecal immunochemical test, have their limitations. Colonoscopy is invasive, uncomfortable, and carries potential risks of bleeding or perforation. Fecal immunochemical test, while non-invasive, may produce false negative results and can be influenced by various factors, including dietary restriction and medication considerations. These drawbacks highlight the need for more convenient and accurate colorectal cancer detection methods. We provide a novel way called NanoCircDx, using circular RNA as our biomarker for detection. Circular RNA, possessing a unique covalently bounded loop structure, has resistance to isonuclease degradation, which enhances its stability within the bloodstream. Recent research has unveiled its crucial role in various biological processes and diseases. Due to its stability and crucial role involved in disease, we selected two circular RNA which has assessed expression in colorectal cancer patients' blood as further detection foundation. So Anne, could you please introduce the detection process for us?
Okay, let me introduce our detection process. In our design of NanoCircDx, we have implemented two distinct pathways for the detection process. In the pathway 1, we will initially employ rolling circle amplification, RCA, followed by the gold nanoparticle-based colorimetric assay. And in the pathway 2, we will perform reverse transcription, recombinant polymerase amplification, RT-RPA, followed by a lateral flow test for the purpose of detection. In the first part of our design, the amplification process, RCA can generate numerous repeats that are complementary to our biomarker circular RNA sequence. Then, the nanoparticles will be modified with short DNA probes, which are designed to detect and conjugate it with the RCA product. In general, when salt is introduced into the gold nanoparticles, it typically induces the aggregation of these gold nanoparticles, causing a color change from red to purple. However, when the RCA product forms a conjugation with the DNA probes on the surface of the gold nanoparticles, it alters the aggregation behavior of the nanoparticles induced by salt, resulting in the color remaining in red. To sum up, a red solution indicates a high concentration of the colorectal cancer's circular RNA biomarker in the sample, whereas a blue solution signifies the absence of the biomarker. In here, our primary objective is to detect the color variation, which is very crucial for assessing the potential risk of colorectal cancer in the user. In the second part of our design, following the isothermal amplification process, RT-RPA, we will use a lateral flow test, PCRD, to read out the nucleic acid, utilizing antibodies on the lateral flow cassette to detect modified amplicons on the RT-RPA product. This detection method yields a straightforward yes or no result for the quantitative analysis.
Currently, we are unable to obtain blood samples from colorectal cancer patients to verify our diagnosis process. We mimic circular RNA by circularizing RNA fragments synthesized from in vitro transcription. To confirm RNA circularization, we performed gel electrophoresis. In this figure, the presence of link 2 indicates the success of in vitro transcription. After adding RNA-SAR to circularization product, any RNA that failed to circularize was degraded. Therefore, the remained band indicates successful RNA circularization. After synthesis of circular RNA, we conducted rolling circle amplification. In this experiment, we conducted RCA using linear RNA and circular RNA sample respectively. Compare link 2 and link 3 with others. Only circular RNA sample can generate low-repeated complementary DNA by conducting RCA. Next, we attached DNA probe to gold nanoparticles for the detection of RCA product. The SEM image confirms the successful synthesis of gold nanoparticles with a diameter of 13 nanometers. After that, we verified the conjugation of DNA probes onto gold nanoparticles by measuring OD value and XPS analysis. Besides RCA, we aim to use RT-RPA to amplify our target. In this experiment, we preliminarily confirmed that RT-RPA can amplify linear RNA and found out the amplification limitation is close to 10 to the power of -12 dilution. We later employed PCRD to detect the presence of RT-RPA product. In this experiment, we verified that antibodies on the PCRD could effectively capture the labeled RT-RPA product and produce test lines, as shown in the figure.
Let me introduce our future work. We initially observed a significant color change in our probe conjugated gold nanoparticle solution when 6 microliter of magnesium chloride was added. In future experiments, we will assess whether the DNA probes on the nanoparticle can detect the RCA product and modify the aggregation behavior. On the other hand, we aim to test whether circular RNA can amplify with RT-RPA. After amplification, we will then utilize PCRD to detect the product.
What a great development! I believe people are curious about the journey of your process now. So, Yvonne, have you inquired about any expert opinion?
Absolutely. That's our human practice member, Christine, to explain this. Hi, Christine.
Since the circular RNA biomarker we are targeting is a relatively new field, we consulted Professor Xin Yingling, an expert dedicated to developing isothermal nucleic acid amplification techniques and various cancer monitoring platforms. Professor Lin noted that the concentration of circular RNA in the bloodstream is exceedingly low, making direct detection quite challenging. As a result, our strategy involves amplifying the circular RNA signal prior to conducting the detection. Considering that the locations outside of typical hospitals often lack precise temperature control equipment required for reverse transcription PCR, we have chosen isothermal amplification methods such as RCA or RT-RPA as our amplification techniques before proceeding with subsequent testing. In order to create visual results that are easy for both human and machines to analyze, we have also adapted PCR screening and the gold nanoparticle colorimetric method. Doctors generally believe that our screening method has advantages compared to current colorectal cancer screening methods. The only issue is how to promote our technology because the underlying principles are somewhat complex. Getting the public to accept and willing to undergo screening is a challenging task. In addition to seeking advice from medical professionals, we were also curious about the perspectives of patients. Through interviews, we learned about the inconvenience patients face when using colonoscopy and the severity of delays in seeking medical attention due to the low accuracy of detection methods. In addition to technical improvements, our team also aims to address the concerns and feelings of actual users.
Then, how do you plan to make this product accessible to the general public?
Our member has considered these problems thoroughly. Hi, author.
Oh, hi, this is Arthur speaking.
And I'm a waiter.
So we are aligned with a total of 5 sustainable development goals proposed by the United Nations. It includes good health and well-being, industry, innovation and infrastructure, sustainable cities and communities, partnerships for the goals and quality education. So let me explain how our research aligns with these development goals and how it aims to help everyone. To begin, our research centers on the early detection of colorectal cancer. Our objective is to foster the rapid advancement of colorectal cancer into advanced stage through the implementation of nationwide screening programs that are both affordable and widespread. We plan to execute this approach in collaboration with the nation's health insurance system. Furthermore, to ensure the widespread adoption of the entire project, Wader will then introduce the hardware device SecMOS and the software app SecMOS-DX that we have designed.
That's right. Regarding the hardware thermostat, we create a fully automated device. It can be controlled with a single button on app ThermoDX. It monitors the entire process, including amplification, heating, and the addition of various solutions. Finally, ThermoDX takes a photo and our machine learning model identifies colors in the envelope. The advantage of our device is simplicity. It can be used even in rural health centers, and a complete test only takes 2.5 hours. No need for complete hospital procedures, expensive machines, or specialized knowledge. If we can introduce thermostats to Taiwan, it can reduce the economic burden on Taiwan's healthcare system. Therefore, thermostats add value to technology products and increase their utility. Moreover, it can be implemented in less developed regions, promoting a positive connection and equality between urban and rural areas. In addition to the medical aspect in Taiwan, we are gradually expanding our vision internationally. So, Arthur, can you tell everyone how our products connect to the world?
Of course, in terms of technological innovation, we have achieved global international collaboration. First, we created the iGEM map to strengthen our connection and knowledge sharing with teams from all over the world. Currently, we collaborate with 21 teams globally, promoting the internationalization of NanoCircDx in technological innovation. Finally, our website offers knowledge related to synthetic biology and general biology, and we have designed our research topic as a game. Our aim is to make it easy for the general public to understand, reaching a broader audience across different age groups, eliminating all inequality in educational equipment. Furthermore, our topic contributes to the sustainable development of the global population, particularly in health and medical development. So, that's how we plan to make NanoCircDx accessible to the general public.
Thanks for a detailed answer. Okay, as you can see, our product, NanoCircDx, has a great potential to revolutionize traditional screening methods, bring convenient, accurate, low-invasive, and affordable colorectal cancer screening method to the public.
The problem of early detection of colorectal cancer seems to be solved by these creative young scientists, NTHU Taiwan iGEM team. I believe people now has a better understanding of colorectal cancer detection. Thank you, Yvonne, for coming to our studio. Again, this is NTHU News Channel. You have just heard the latest news around the globe. News with no bias, news with no exception, delivering nothing but the truth. This has been Kira and our guest, Yvonne, bring you the latest news around the globe.